Kia ora and welcome to today's LINKOnline webinar. Myrtle Rust is an invasive disease that has huge implications for our native ecosystems. And as we build understanding of the disease and its spread, we're also considering how to apply these lessons to other potential invasive pathogens. Research and control is happening across multiple organisations and agencies. Today we hear from Marge Pādamsey from the Beyond Myrtle Rust Research Programme and Maureen O'Callaghan from Ngā Rākau Taketake, New Zealand's Biological Heritage National Science Challenge, who will provide a shorter overview of work in progress. James McCarthy will then hold the floor with his fascinating work on refugia. I know you'll have lots of questions. Please put them into the chat box early and we will put as many as we can to the panel. Rest assured, even if we cannot answer today, we will post Q&As to our website in the next week or so. And now handing over to Marge.
Kia ora koutou. I just want to introduce the Beyond Myrtle Rust program briefly. Beyond Myrtle Rust Towards Ecosystem Resilience is a 5-year endeavor program that's going to continue for another 3 years. And the research is framed around 4 questions. How the pathogen Austropuccinia psidii is adapting to New Zealand's environment. So this work is led by Stuart Fraser, who's at Scion. And also has input from the University of Queensland. The second question is how are our ecosystems being impacted by Myrtle Rust? And this is work that's being led by James McCarthy and myself at Manaaki Whenua. And we have input from the University of Auckland, from Unitec, and from Plant and Food Research. Our third question is how can we control or mitigate the impacts of the pathogen on our ecosystems, and this is work led by Grant Smith at Plant and Food Research with input from the University of Canterbury and Lincoln Agritech. Our fourth question is how can we enhance kaitiakitanga and Maori-led solutions in pathogen-affected ecosystems? This is work being led by Albie Marsh at Plant and Food Research. So with that, I'd like to hand over to Maureen.
Kia ora koutou. I'm just going to really briefly introduce you to another research program that's focused on Myrtle Rust that's really closely aligned to the Beyond Myrtle Rust program that Marge has just outlined. So Naraku Taketake, or Saving Our Iconic Trees, is a science investment fund program led by BioHeritage Science Challenge, and it has the aim of developing new solutions for both Myrtle Rust and curry dieback. And the research program's been co-designed with Manaaki Whenua researchers and stakeholders from a wide range of agencies and organisations, and it addresses the science priorities that were identified by the Myrtle Rust Science Advisory Group. So next slide, please, James. So NRT has 7 research themes, and the titles of them here are quite self-explanatory, but the research includes Maori-led projects, predominantly in Aotearoa, but also happening in all of the different themes. We're looking at social dimensions to forest health and driving what underpinning values and so on drive mobilising, getting communities mobilised to take action. There's new tool development going on for management of Myrtle Rust and understanding more about the host pathogen and conservation priorities. Next slide, please, James. And this is just gives you a bit of a flavour of the research that's underway. So, for example, in Aotearoa, led by Te Tira Whakamātaki, there's a whole lot of a suite of programs going on that are Maori-led, and one of the really important ones that's a focus at the moment is developing best practice protocols for germplasm collection and protection. And I'm not over the next subsequent Myrtle Rust webinars, we'll be talking about some of the projects that are within these in a bit more detail. But in the meantime, there's our website up the top there, and if you have any questions, you can refer to the website or contact us through that. So I'm going to hand over to James because he's the star of the show today. Thanks, James.
Thank you, Maureen and Marge. So yes, my name's James McCarthy. I'm a plant ecologist at Manaaki Whenua Land Care Research, and I'm going to be talking today about some work that we've been working on for the last couple of years. First, the idea was to try to map the distributions of New Zealand's native Myrtaceae, and these are species that might be susceptible to myrtle rust. And then some subsequent work was to see how much of those populations actually occur outside the disease range. So how threatened are certain particular populations and how much of these populations are likely to be impacted. So a lot of people involved in this work, some people from Manaaki Whenua, but also colleagues from Plant and Food Research and NIWA. So I'll just start off with a bit of an introduction to myrtle rust. It's caused by a rust fungus called Austropuccinia psidii. It's native to South America. It's a wind-dispersed fungus, and often the symptoms are these yellow or sort of gold pustules that you'll see on leaf surfaces, but also on newly formed stems and fruit and flowers. It only affects new tissues, so it's often seen when plants are putting on new flush of growth. One thing that's strange about this fungus, about this rust, when compared to other rusts, is that it has a really wide host range. Normally rusts are really specific, so in this case, the affected species are confined to the Myrtaceae family, of which there are about 6,000 species worldwide, and at the moment we know of about 480 confirmed host species, which is a lot. It's got a pretty wide distribution these days. Originally, as I said before, native to South America, where infections were usually pretty mild on native species. So the native Myrtaceae in South America had sort of co-evolved with this fungus, so there weren't really huge outbreaks. People knew about it, but it never really caused huge harm until introduced Myrtaceae started being brought in. So these are species like eucalypt trees that were planted, and these were more heavily affected. So in the 20th century, 1900 to 2000, it started spreading around a little bit in that area. It was detected in the Caribbean in the 1930s, then Mexico in the 1970s, up to North America and Florida in 1977, where it primarily affected an exotic Australian bottlebrush, which is a pretty bad weed there. It also went to California at some time in that time, but since 2000, that's when it's really started moving about, coming sort of a bit closer to our corner of the world. It was detected in Hawaii in 2005, Japan 2007, southern China in 2009, and then a big one, it arrived in Australia in 2010. And this was bad news because Australia is a real hotspot for Myrtaceae diversity with thousands of species. And there have been some pretty heavy effects of myrtle rust there. New Caledonia in 2013, Sumatra in Indonesia in 2015, Singapore 2016, and then in 2017 it was detected in New Zealand. Initially on Raoul Island in the Kermadec, which is well north of the North Island here, where it was detected on the Kermadec Pahutakawa, Metrosideros kermadecensis. And then shortly afterwards, in May 2017, it was found in a nursery in Northland, and it spread from there. So MPI initially tried to contain the disease with hopes of eradicating it, and many infected plants were removed, mostly planted ramarama, which is Lophomertus species in their hybrid, a picture of which I have on the right here, which is affected by myrtle rust. And I was just out in Taranaki last week with some other colleagues from the Beyond Myrtle Rust program, and there are even tracks that are being closed due to the myrtle rust infection. Not too dissimilar to what we're seeing for Cody dieback, but it's probably beyond that. Well, it is beyond that the eradication sort of phase now, and there's a lot of research going on just to find out how much impact this disease might have on our natural systems, especially. So in New Zealand, we have 27 native species of Myrtaceae, all of which except Manuka, Leptospermum scaparium, are only found here. So there's an exceptional amount of endemism in the family. Most of the species are in the Metrosideros, which include Pahutakawa, the ratas, and a bunch of climbing ratas as well. Also, Kānuka, of which there are about 10 species now after a revision in 2014. 2 species of Lophomertus, and they're hybrid. Then Manuka, 1 species, and then there's a species of Neomyrtus and a species of Syzygium. And there are also about 200 or over 200 introduced species of Myrtaceae here in New Zealand. These include Vajoa, eucalyptus species, bottlebrush species, and others. So back in 2017, Manaaki Whenua was contracted by MPI to produce some maps predicting the distribution of these native New Zealand Myrtaceae. So to create these maps, we use species distribution models. So when you think about a species like Pahutakawa, which I have in these pictures here, you think about a species that usually grows on the coasts in warmer northern areas. At least that's what we know about its natural distribution. And this is the kind of information or data that we use to make models that predict a species distribution based on the characteristics of the environment from which we know it occurs. So using plot data from the National Vegetation Survey Data Bank, we know that Pahutakawa is found in these red locations here. As I mentioned before, warmer sort of northern coastal areas, and that it's absent from all of these locations. Now you might wonder why we'd want to put a map together showing the distribution of a species if we already have all these point records. These point records would be very useful if you wanted to take a photo of Pahutakawa, or maybe if you wanted to go and collect some samples from it, or some leaf samples or something like that, or some seed. But what you don't get from this kind of information is where else does it occur? Does it occur in these areas between the points, for example? Also, we know that it occurs further south along the coastlines than these points here show us, and that's not always picked up in plot data. So with these presence and absence point records that I showed you before, we put these into a model which builds relationships with a bunch of environmental variables. In this case, we used a modeling type called boosted regression trees. And these models can then be used to predict a species spatial distribution using environmental variables that we do have some sort of knowledge about for all of New Zealand. So here's an example of the prediction for Pahutakawa. Now, Pahutakawa is a bit of a special case because I'm sure many of you know that it does occur as far south as sort of Sumner and Aotearoa near Christchurch, where I am. And it's also commonly planted on the Wellington waterfront. But for this exercise, we're interested in the natural distributions of Myrtaceae. So that's excluding planted individuals. And as you can see, it fits pretty well with the point records here in red, and also produces a distribution that predicts a little bit further south down both coastlines like we had hoped. So distributions were produced for almost all of the native Myrtaceae. The eagle-eyed among you may notice that a couple are missing. We didn't have enough distributional data to produce models for Bartlett's rata or rata moiho, nor the Kermadic Pahutakawa and Metrosideros carminia. And some of you may also know, well, as I mentioned before, the taxonomy for Kānuka was recently revised, where it was split into around 10 species. So for the purposes of what I'm about to talk about next, we worked with the previous lumped taxonomy. But if any of you come across the report that we produced for MPI, which is available online, you'll see that we did produce some distributions for the new species of Kānuka, but we were using a different modeling technique that didn't fit in with what we were doing here. Now, most of the distributions seem to make sense. Manuka here, for example, Leptospermum sciparium has a really wide distribution from the far north to the south in Stewart Island. Also, some of the ratas, Metrosideros umbellata, which is southern rata, has a mostly southern distribution. Northern rata, Metrosideros robusta, mostly northern, as you'd expect. So we think the models performed quite well. And in a paper that just came out last week in Journal of Applied Ecology, we use these models in conjunction with what we know about the range of the disease to identify areas of refugia. So these are areas of Myrtaceae or potential host distribution that sit outside the disease range. So I've got a bit of a figure showing that here, where the yellow area signifies one potential distribution for the disease, and then all of these little blobs, so this dotted blob here, these two green blobs, and these blue blobs are a hypothetical distribution of a species, of which some is inside the disease range here, this dotted line, and the rest is outside. And outside, you've got green areas, which are currently forested, so in a land use that would support the species. And then the blue areas are in some other land use. So we know that the environment is probably right for the species there, but we know that it won't be there because it's currently the area is currently being used for something else. So this work that I'm about to talk about now was funded by the Beyond Myrtle Rust program that Marge introduced, as well as some of Manaaki Whenua's internal funding. So in this paper, we presented results under 2 myrtle rust scenarios. The justification behind those is a bit complicated, but the likely reality probably sits somewhere between these two, because we do know that it occurs around Wellington and also in Marlborough. But the results I'm presenting today will be from scenario 1, which is the one on the left, the more optimistic of the two, which we think probably captures a more sort of core range of the disease where effects might be more severe. And the work behind predicting these myrtle rust maps that I'm showing you now is detailed in the paper in the bottom right here by Rob Beresford and his colleagues, and this was published in 2018. So here we can see in the red the predicted distribution of ramarama, or Lophomertus ballata, which is a species that we know is heavily affected by the disease. I apologise if it's difficult to see, but a decent amount sort of up here in Northland, also extending down the west coast of the North Island, a little bit in East Cape, and it's tricky to see here because this figure was modified a bit for the figure for the paper, but we also did predict it in Marlborough. So we predicted about 614 kilometres squared of range for this species, of which only 15 kilometres squared, again, sorry, it's difficult to see, but I'm sort of hovering over these little blue areas here. So only about 2.5% of its range sits outside this disease scenario here. But when you think about areas that are environmentally suitable but not currently forested, you get about another 129 kilometres squared. So there is a decent amount of area that can be restored to provide additional refugia for this species. And so basically, with a northern species like this and a disease that also grows in warmer areas, you're going to have more of those ranges colliding, I suppose. But when you take it from the other end, southern rata, Myrtaceae umbellata, has quite a wide distribution, about over 11,000 kilometres squared, we predict, mostly in the South Island, also extending north. About 90% of the species refugia, range sits outside this disease scenario here. So we don't actually have much information about whether southern rata is likely to be susceptible to myrtle rust. It may not be, but even if it is, we can take a bit of confidence from the fact that a lot of its range will at least sit outside the area that the disease will be at its worst, especially. And since Myrtaceae umbellata is generally sort of confined to areas that are more intact, so the west coast of the South Island, there's not too much restorable refugia for this species, but there is a bit in the far south and also some patches along the west coast there. And sorry to put a table in a talk, but I just wanted to show this last sort of piece of information here. These are the species that we did this exercise for, ranked by the percentage of refugia that they might have. So this is the area that sits outside the likely disease range. As I mentioned before, Myrtaceae umbellata, which is southern rata, is right at the bottom with about 90% of its 90% of its range sitting outside the diseased area. And then at the top here, we've got a few that we perhaps should be concerned about. So there's Lophomertus ballata, which I showed you before, about 2.5%. This is really a species that we need to. It's probably not going, if it is really heavily affected in the wild, it's probably not going to survive in refugia. And if it does, there'll be a huge bottleneck. So it's species like these that we need to start thinking about either disease control or perhaps other methods of conservation, like seed banking and botanic gardens, and perhaps planting outside their natural range. Others that don't get thought about too much are Syzygium myri, Mairi tawake. Only about 0.6% of its range sits outside the disease range. And then worst of all is Myrtaceae albiflora, which is a climbing rata. Not commonly sort of considered or come across in the field even. Only 0.1% of the species predicted range occurs outside the range of the disease. So this really is a species that probably deserves some attention. We need to work out whether it is likely to be affected, because if it is by Myrtle Rust, because if it is, it's unlikely that it will have many areas that it can escape the disease from. What's next? So ground truthing the predictions. These, of course, are models. These distributions are models, no sort of substitute or surrogate for people on the ground and local knowledge. And so anything that's implemented as a result of the results that we've found here would, of course, need to be ground truthed, especially at regional or local scales. So these maps were put together at a national scale, and so they're probably likely to be broadly right and precisely wrong. So a bit of ground truthing would really help. Improving the data, plot data, the plot data that we used to put these models together were extracted from the National Vegetation Survey Databank. We're always keen to see more data go into the National Vegetation Survey Databank, because then they're on hand for us to use at short notice for projects such as this. Incorporating refugia into reserve management. Some areas of National Park, for example, might be more Myrtaceae diverse than others, and might be good areas to keep an eye on and just make sure that they're in good condition. Thinking about native restoration opportunities, 1 billion trees, can it be worked in alongside this to provide additional refugia for affected species? The distributions could be used for targeted seed collection to better sample the genetic diversity of a species, and then also some species that are basically entirely captured by the range of the disease itself. Ex situ conservation could be might be a really important option for these, and perhaps I've heard from Australia terms like rescue gardens, where particular species, entire genotypes are planted as sort of a reserve population in case things do get really dire. I'll leave it there with my acknowledgement slide. I'll thank MPI and Beyond Myrtle Rust for funding this project. Some people helped with data access: Elise, Aaron, John, and Vijay. Liv, Tom, and Jane helped with modelling advice, and then Larry, Alex, and Channel helped check some species records early on in the project. That's me. Thank you.
Thanks, James. Thank you, James. That's a really interesting and fascinating talk. I think there are lots of questions coming in. So I'm just going to start with hopefully an easy one. Is Myrtle Rust intolerant of cooler conditions?
It definitely grows in warm areas and humid areas. So those are the areas that, as you would have seen in those Myrtle Rust maps, northern kind of areas. So yeah, it's less tolerant of cooler conditions. That's right.
So leading on from that, how might climate change affect the predicted distribution of Myrtle Rust?
Well, it's predicted to get warmer. It's something we've thought about, but not something we're sort of actively working on, unfortunately, at this point. It's tricky because you've got two moving parts, as I've sort of shown here. You've got the range of the hosts and you've got the range of the disease, and the disease can respond more easily than the host. So if you have climate changing quickly, then a rust that moves about on the wind can find its new climate space more easily than a plant, especially a woody plant like the Myrtaceae. So with climate change, I would expect, well, we know that New Zealand's going to get warmer. What's going to happen with humidity is more difficult to pin down, but it almost certainly would result in some sort of change for both the host and the disease. But how exactly, I'm not quite sure, because it's a little bit difficult to sort of disentangle, but it is something we're thinking about.
Question for, I think this one's for Maureen. How many species have, Myrtaceae species have good representation in our seed banks, or in have germplasm stored for safekeeping? Is that in your program?
I can't answer that question, but we could find that out and include that later on. Probably not very many, I would say, and the storage conditions may not have been right for the seeds that have been stored to date.
I know that Doc did a big effort early on to do some seed collection for seed banking. I imagine that it might have been quite complete. Some species are difficult to store in seed banks, especially fleshy fruited things. So these would be our sort of rainforesty kind of, I think, Syzygium Mairi Tawake, perhaps the Lophomertus species. This is probably a question we can answer offline, but I know that there have been some efforts to put some to do some seed banking, and there is part of the Ngā Rākau Taketake project, the restoration and conservation part, does include some seed banking research. I'm not sure exactly what it is. I don't know if Marge, if you've got anything to add.
The Naraku Taketake research in the seed banking and germplasm space is starting with developing culturally acceptable processes and for collection and protection of the germplasm. And once that's in place, there's also some work going on with looking at improved storage methods. It's a work in progress.
Great. So we'll be sure to get a little bit more information together to pass on to you about that. Now, early on, somebody asked a question that was one of the things on the top of my mind as living here in Wellington. Why is introduced Pahutakawa in areas, you know, like Wellington, not important to study? James, can you help us with that?
They are important to study, and we do think about them. In this case here, we were thinking about our natural forests. I suppose we weren't thinking about areas that are planted. Areas that are planted tend to be areas that are more frequently visited by people. So along the Wellington waterfront, I guess you could be relatively confident that if there was some sort of Myrtle Rust outbreak, it would be detected by somebody. So I guess with this study, we were looking to try to think about what might be happening in natural areas that are less frequently visited, but yeah, not discounting these planted individuals at all.
That's good to know. Two more quick questions, maybe quick. Should the threat status for Manuka be revised based on the information that you have from your research?
So Manuka, well, following the arrival of Myrtle Rust, all of the Myrtaceae were moved into one of the threatened categories. I don't know exactly what it is. So I guess you mean revising out of that category. It's difficult. There is a lot of work going into Myrtaceae at the moment to see whether it is affected. I think some preliminary results show that it is in the lab, but results in the field are variable. So work is still going on, and it's probably too early really to make any sort of revision or to know confidently how affected Manuka is. That's my understanding at least.
Right, and I think we'll squeeze in one more. Considering the wind-borne nature of dispersal of the pathogen, would refugia, particularly in the northern parts of the country, help protect host plants locally?
Well, yes, I mean, I'd say so. So I guess the climate doesn't stop the spores from going somewhere. It just stops it from, I guess it stops it from growing once it arrives there. So this thing can get all over the place. It made its way here from Australia across the Tasman. So they probably are quite abundant in the landscape really, and it's just a matter of it finding a suitable host in a suitable environment at which point it would grow. So yeah, it really is about that environment, and so I think the answer would be yes.
Great. As usual, we've got more questions than we can answer. Thank you very much for sending those in. You will hear from us. This is actually the first in a dedicated series of webinars to share research and solutions for Myrtle Rust, and Renee Johansson will be in touch with today's participants with invitations to future events in that series. And Renee is just waving to you, I think, at the moment. We will return with a further LINKOnline webinar next Tuesday. The topic is soil health. Watch your email for the invitation, and we look forward to seeing you again next week. Ka kite anō.